Hello, and welcome to the Activation Nation podcast, your weekly source of actionable advice from industry experts in self-development, health, LifeVantage products, network marketing, and more to help you activate your wellness, your business, and your life. Today's episode focuses on product, one of the 4 pillars of belief. But first, the legal stuff. You may hear our guests talk about the income they've earned or how their health has been affected with LifeVantage. Please note that the average annual earnings of a typical active LifeVantage distributor in 2021 was $704. For the most up-to-date information, please click the link in our show notes. Any product statements have not been evaluated by the Food and Drug Administration. Our products are not intended to diagnose, treat, cure, or prevent any disease. And now, here's your host to dive into today's story to help you activate your life.
Sorry, guys, I'm laughing. Welcome to the show. We're so glad you're here. You know what? A couple weeks ago, we did this, and we had some technical problems, and so we're having a little fun with everybody here this afternoon. My name's Jesse Allen. I'm the Senior Vice President of Product Marketing here at LifeVantage, and 2 of my favorite people, not just even at LifeVantage, 2 of my just favorite people are here today. Lisa
Sure.
Barnes.
We want that in writing.
I'll write it right now. Lisa Barnes, our VP of R&D, and Christina Beer, Dr. Christina Beer from the R&D department as well. This is a continuation. If you were with us a couple weeks ago, we talked all about product efficacy, product safety, product quality, a real emphasis on what we do to ensure those 3 aspects of the products. And this is, we talked about it a little bit, that this was kind of a 2-part. This is a continuation of that conversation. Unfortunately, Adam's not with us, who was with us last time, our Director of Quality, but Christina's here now.
Thank you.
And we're going to talk all about the clinical studies and the studies behind our products, behind raw materials, different types of studies. I think we got 3 different types of studies that we're going to talk about, and why we use one versus another, their purpose, and all about all that. Right?
Yep. That's the plan.
Did I miss anything?
You got everything. No. You did well.
You got it right on target.
Thank you. You guys were off to a good start.
That's why we're one of his favorite people.
We're off to a good start. Okay, here are, just to set the course and a bit of an agenda, there's 3 different studies primarily that we're going to talk about. In vitro and, can I say in vitro and RNA sequencing?
Yep. RNA sequencing is an example of in vitro.
Okay. Then we're going to talk about like consumer trials. So, and then we're going to talk about human clinicals. Right?
Correct.
And are we going to keep this all around a finished product, or are we going to also talk about those tests in conjunction with raw materials, or is it we're just going to piece it all together?
We can piece it all together, but probably focusing more on the product side, since we did cover a little bit of the raw material last time we chatted.
Okay, so
because that was partially how we knew what ingredients to choose, is that we looked at what clinicals had been performed on them, which actually are some of these different categories that we'll discuss.
Okay.
So we can segue in.
Perfect. Well, let's get started. Let's start with in vitro, and you might need to define that so people know what we're saying when we say in vitro studies.
I'll let you take that one, Christina.
Sure. So in vitro is a fancy Latin word for doing something in a cell. And basically, you take the cell from some kind of organ that you are interested in for a absorption study or a metabolism study, or, just depending on what kind of study you're looking at, you choose a certain cell line. So you grow up the cell, and then you treat it with our product, the product we're interested in, and you get a response in that cell. Right?
Yes.
And depending on that response, it tells you what we're looking at. For example, if we do our Nerf2 product, Protandim Nerf2 Synergizer, we take the actives, we grow the cells. So,
and what cell types do we typically use?
Yes, exactly. So we want to use a cell type that turns over quickly because it's metabolically active. For example, a liver cell or a kidney cell or a heart cell or a gut cell, because the turnover of those cells in the human body is quick. So we want to study the effect of Nerf2 in those cell types. Mm-hmm. So then there's a method, and I'm not the expert. We use third-party experts to grow the cells. There's a certain way of growing them, certain media and conditions. And then we treat them with Protandim, Nerf2. and then we read gene expressions, most likely. Or we can do protein activity assays that we're looking at. Or we can do protein analysis amounts, quantitative amounts, what that cell produces under that treatment.
Can we let's back up just a little bit, and because you mentioned third party, and I think that's important that all of our studies, well, the majority of our studies, maybe not the consumer, but certainly in vitro and human clinicals, they are actually done and performed with a third party lab.
Correct.
And maybe without going into too much detail and just boring everybody, not that it's boring, like our IRB. And what that means. And the reason I want to do that is because I want to help understand and create this narrative of, like, we're not just sitting at a desk and it's just a couple of us that are doing this, but there is protocol that is followed, and that help us understand that.
So IRB is an internal review board, and that's usually, you have to get an approval if you do a human clinical, which we'll talk about a little later.
Okay.
So for in vitro studies, you don't need an IRB approval.
Because we're not testing on people.
Yep.
Right.
Okay.
So when we do and when we decide to do an in vitro study on one of our products or even a raw material, then we have a third-party lab that they're experts in this.
Yeah.
We're not experts in this. We have the theory. We'll write up the protocol with the experts. We tell them this is what we want to study. And then there's a dialogue going back and forth. It can go on
What's the best
for
way? And that's
2 weeks.
Yeah.
What's the best way of doing this? And then we listen to these experts, and then we come up with a protocol that we can agree upon.
Yep.
Yeah, they're very used to us giving them a call and being like, okay, so we have this crazy idea.
Yeah.
What's the best way of making it happen?
Yep.
And so then we get on a discussion with them. We say, okay, this is the gene expression we want. What cell line should we do it in?
Yeah.
What should be step 1, step 2, step 3? Because you can't just go right in and just pick any sort of cell line and just start dosing. There's multiple steps you have to do. The first thing we do with any of our in vitro studies is we have to determine what dosage we use, because it's not going to be the dosage that we're having exactly in a pill.
No, and
It needs to be appropriate for the cell type, just so we don't.
Or we're going to kill it.
Okay.
Right? We're going to overdose and we're going to put too much stuff in there.
Exactly.
And we're just going to kill
Nothing
the
to do with
cells.
product safety in a
No.
human, but just specifically for one, like
Yeah.
a cell line.
And so right, a cell is grown. So we've got a product let's just say we've got a product that is going to address heart health.
Yes.
Mm-hmm.
So they take a cell or somehow they grow heart cells.
Mm-hmm.
Then literally in a dish, right?
Yes.
They take the cells that are in the dish and then they inoculate or expose those cells to
Different dosages.
the dosages
Right.
of what would be the equivalent of the finished product that an individual would be
Correct.
consuming.
Correct.
And then they measure how do those cells respond to the dosage that was applied to
Yes, not just dosage, but we do it over time too.
Uh-huh.
Because every gene expression in a cell is different.
Yeah.
It expresses at different times. So
Yeah.
we have different time points where we actually take measurements as well.
And the end objective, what's the end objective of doing that? Is it
So the end objective is to have the optimum dosage for the right amount of time so that we have the best expression or the best reaction of this gene in this cell line.
okay? So we look at different dosages and then and that helps us so this is done during the development process to help us identify that we've really formulated the right way and that we are providing the most beneficial end product to the consumer.
Correct.
Yeah.
It's stage 1 of our testing. And so then
Okay.
once we've determined that correct dosage and the correct time points that we want to explore, then we really need to look at what are our objectives there. We can look at specific gene targets. So if we already know we want to look at gene A, B, and C, and that's what we're really interested in. I don't really care what anything else does. I really want to see what it does in these 3 genes. That's kind of a more targeted approach. And sometimes we do that with NRF2 if we're looking at new ingredients that could potentially activate that pathway, because we know what some of those hallmarks should be. So we go very targeted. One benefit of going targeted is that it can be quicker, and a lot of times it can be more inexpensive. So if we want to just test a whole bunch of different materials, that's a good screen through.
Okay.
Also, we can go more exploratory and go broader. And that's where RNA sequencing that you mentioned comes in, where you're really looking at the entire genome of whatever cell type that you are investigating. So every gene that'll be expressed, upregulated or downregulated, you'll see all of them. So essentially what you'll end up with is rows and rows and rows and rows, tens of thousands of rows of genes, because there's so many that are expressed in each cell type, and you can see the impacts and the significance that your product had on all of those. The beauty of RNA sequencing, so gene expression is very good at targeted. Let's see exactly what's happening. The RNA sequences is more pathways. So we see all these genes that are kind of clustered together and have similar functions. Okay, let's kind of trace how it's working, and we can understand the mechanism of action for it.
You mentioned like a couple of weeks. Like what's the time duration for like an in vitro study?
So
on
average, ideally it can take 2-3 weeks to get the protocol ironed out so that we're happy, both sides are happy. And then in the meantime, since we have a history with this third party, they already order ingredients, they know what's going to happen. And then once we get the go ahead and do the study, it can take anything from a week, depending on how many genes we want to look at, to a month for results to come in. Right? But then comes the harder part, and that's analyzing the results.
Yeah.
Right?
Exactly.
To your point, right? Like 10,000 or
yes.
thousands
Literally,
upon
not
thousands
even
of rows
exaggerating.
of data,
Yes.
And all of that has to be analyzed to figure out what is it really doing.
Correct. And there even are calculations that need to be made to understand the significance of the data and determine, for example, if we're looking at this gene, what fold increase or decrease did we see? It's not laid out easily for that.
Yeah.
It's just data. And so there's math that needs to be done for every single row. And so Christina we've been seeing it on a study we've been working on currently, where she's just been digging into her hole for hours at a time.
Yes, I put the blinders on. And don't bother me. And I have tunnel vision.
And I'll come over and talk to her.
I dream about numbers.
Exactly. She's just like, I'm just seeing rows of Excel.
Okay, so a couple of weeks to a month to like write up the protocol and the back and forth with the lab and how really should we be testing this?
Exactly.
And what should we be looking at? Right? So we do that, and then a week to a month of actual inoculation, and then we get the data back, and then it's really just
however long.
Yeah, based on the amount of data. So it's not insignificant, right?
But it's much faster than any other forms of testing, which is another benefit of it. So you get kind of this speediness to getting results. You can screen through things quickly, less cost, and then we don't need to go through all of the necessary approvals like our IRB, which we'll talk about. So there's a lot less barriers to entry. So we can really get a lot of data fast.
Yeah. And you mentioned, you just said cost. Like, give me an idea of what an in vitro study costs on average.
Well, I would say, depending on how many genes they want to analyze, because there's costs associated with every gene, because you have to make a prime. It's all technical. So I would say it can run anywhere from $20,000 to $60,000, $70,000, $80,000.
Okay. So it's an investment. Yeah, it's an investment for the organization to do that.
And you can even do some more targeted ones that are just, if you test 2 products and maybe only a handful of genes, you can even get it down like closer to 10.
Right.
Yeah.
We've just been doing a lot of bidding, so I have like a PDF that's in my mind of different tiers of studies.
Right.
Okay. So that then tells us what we really end up with once the in vitro is done is it tells us how genes are being upregulated or downregulated. Correct. Which gives us an indication of how the benefit that it's going to provide to the end consumer, right?
Correct.
Because the gene, their cells are going to respond more or less the same of what we saw with the in vitro study, right?
Correct.
Genes are going to upregulate or downregulate, and we know that when genes are activated one way or the other, there is a benefit or a response to the organism or the human. Okay. Then where do we go from there? So we've done an in vitro study. We've seen we have identified which genes are activated and moving which direction. We have satisfied or proved that, okay, this is what we thought was going to happen, this is what we wanted to happen, we now see and have proof that it is happening, then what's the next step or the next study?
Then you got to put it on people if it's a topical, or put it in people if it's an ingestible. You need to let them try it and make sure it has those benefits that you saw in vitro actually in their lives. And there's two different ways that you can do it in people.
Okay.
First, you have the consumer study.
Yep.
And then we also have the clinical study that we mentioned earlier. So consumer is a little easier and, once again, less expensive most of the time. They can make it pretty. We've seen some expensive quotes for them. They can run high too. But it tends to be less invasive. You're not drawing any sort of blood. You're not analyzing any fluids. You're mostly just going through questionnaires and really understanding the consumer experience. So they're using a product for a month. And then you're doing a periodic questionnaire. You have baseline, 1, 2, 3, 4 week. Did you see change? And really draw out the benefits from there. It's more subjective, but it really, when you think about when you're creating a product for someone, I mean, objective measures are wonderful, but if they're not feeling it, if they're not experiencing the benefits, then are they going to keep buying it?
Yeah.
Like, we want you to, if you're experiencing something, we want you to feel better. And so we want to understand that consumer experience piece. I mean, beyond efficacy, during a lot of that, we get input on the format, the flavor, the experience, any potential side effects. We hear a lot from that helps guide any additional steps. Do we move forward? Do we go back to the drawing board? It gives us a lot of information.
Yeah. Okay. So with the consumer trial then, or the consumer study, we're looking at, we're measuring multiple things,
Correct.
right? Because it is subjective from the trial group, and what from like a, and maybe this isn't the right way to think about it, but from like a statistical significance standpoint, like how many consumers do we typically want looking at the product? Because opinions of two doesn't make any sense.
No.
So how many people do we typically have in those consumer trials, or what's the ideal maybe?
If we're looking at a topical, we want to have at least 30 people.
Okay.
To get somewhat of a difference in opinion and actually get a statistical significance. So the amount of 30 people is usually what we go with. For an ingestible, we want about 50 people, hopefully. That would be the ideal for a statistical significance. But then it also depends on what are we looking for, right? If it's just exploratory and to see if are we going in the right trend, or is this not working at all? So I would say 20 people is all we need just to see a trend.
Yeah.
But it won't be statistically significant, that trend, but at least we see that we're going in the right direction.
And you could follow it up with something else.
Exactly.
Something also important to note between topicals and ingestibles is how we approach the study. So with an ingestible, we always compare it to a placebo.
Yeah.
So we have just a capsule that's filled with filler, and it's just, you just take it and then compare to our active product. For topicals, we don't do this. We don't compare to a placebo. The raw material suppliers, when they develop their materials, they'll do their clinical studies versus a placebo that doesn't include that ingredient. And that's great data.
Yeah.
But for topicals, we don't think about topicals as just one ingredient in a delivery system. The delivery system is part of the activity.
Okay.
You get so much benefit from hydration or from antioxidants or some of these just kind of base pieces of your formula that you wouldn't consider your actives. So your entire formula is active. I actually got in a huge fight about this with one of my previous job with someone who only did ingestible clinicals. And he's like, you always need to have placebo controlled for topical as well. And I'm like, I consider my entire topical formula to be active. And
Got it.
we always test versus untreated. Because the minute you introduce something to your skin, you will start seeing some benefit.
Yeah. That makes sense.
Yeah.
So maybe a plug for those that are watching or those that are listening. The consumer trials we do on a fairly regular basis.
Yeah.
Yes.
And we are always recruiting consultants, right? LifeVantage consultants to actually participate. Not always, but quite often we're recruiting them to participate in the consumer trials. So if you want to participate, comment, right? Just let us know. We'll add you to the list, or email us. And I always forget product questions.
Yep. Product
with
questions.
Product questions at
LifeVantage.com.
LifeVantage.com. You can email that and say, hey, I want to participate in a consumer trial, and we'll add you to the list.
Yes. And with the consumer trials, we have two ways that we do them. So the list that you would be on would be for our internal consumer trials, where we'll kind of field the study, we'll gather it, we'll do the statistics. We also will sometimes employ third parties, especially depending on the scope of the study. If there's going to be quite a few participants and the statistics are going to be pretty complex, we'll use a third party. But the cost obviously does go up exponentially with that. So we'll do both models.
And I don't think we necessarily always do it this way, but I think the majority of the time we will do like an internal study, get a pretty good idea, and then if we need to use if we want to use the information for marketing or whatever, then we'll use that third party, right?
Correct. Exactly. Just for that additional validation.
Yeah. And it removes any subjectivity of
any
us.
bias.
That's right.
Yeah, any bias. That's probably the right word. Okay, so consumer trial, and sometimes we will end there, right? Or the consumer study. We won't go on to a human clinical.
Agreed. It depends on the product type.
Talk to that. Like, why would we make the decision to say, okay, we're satisfied with just a consumer trial. We're satisfied there. We know we've got a good product. A human clinical is not necessary. How do we make that distinction?
So for me, when I think about it, with a product that's incredibly experiential, the most benefit that I get from a study is from this consumer trial, because we're getting really the opinions of them. Some of the objective things are okay, but I don't think it adds as much benefit. So if the product is very experiential, then consumer trial is usually it for me. For some of the more higher anti-aging, potentially, but there's a lot of cost that comes along with it.
Yeah, for example, if we do hypothetically, we would make a joint product or something like that. We don't have the expertise to evaluate a joint health because there has to be inclusion criteria. You're not eligible because your joint is too far gone. You're included because you're right in the right target of where a supplement would be beneficial. So there's a lot of inclusion and exclusion criteria that we don't have the expertise or the instrumentation to do.
Instrumentation is huge.
Yes, and therefore we have to use a CRO, which is a clinical research organization, and partner with them, and they will write the protocol with our help and our approval. They will get the IRB approval so that we can actually do x-rays or whatever it is that you want to test, and then they run the whole study, write the report, and send it back to us.
Yeah. Okay. So the reason to maybe end with at consumer trial is going to be product specific.
Correct.
Exactly.
What we need to measure.
And kind of what claims we want to make. For example, on the skincare side, if we're okay just making claims like X% of people agreed that their skin looked better, then this consumer test, perfect. That's what that information gives us. And if we have enough people in the consumer trial, we can say that with confidence.
Yeah.
But if we want to say we saw an X% increase in hydration, that needs to be done with instrumentation. And that's where you need to go to a clinical. Or we want to have a dermatologist grade it. Because even though they're assessing it with their eyes, we don't count that as subjective. They are trained and they are honed in. And so the dermatologist grading is actually an objective grading. Yeah. They know, they've seen so much. They can grade it on a scale of 1 to 10, and it can be treated as a kind of like instrumentation because they are finely tuned. And so it kind of depends on what we feel like is going to be the most compelling for the certain product. Once again, product specific, but there are some metrics that it does make sense to bring in instrumentation. For example, if we want to say that we saw an increase in hydration in 24 hours, third party, that's a quick third party human clinical, because once again, just 24 hours, take it one or put it on once, come back in 24 hours, done, then just do the analytics with it. But they can go from all the way from 24 hours out to 12 weeks plus for some of the anti-aging measurements. But it does add the objectivity that come from those different metrics.
Okay, let's talk a little bit more. Give us the rundown of the a human study and And I mean, you talked about it a little bit already, but how it works, what's the protocol, and why we would choose to do that.
Okay. Well, what we will start with is talking with whoever our contract research organization is. And maybe even, for example, for skincare, we just talk to a dermatologist who can sponsor the study.
And some CROs are specialized in certain areas.
Agreed.
So you need to find the right CRO for that.
So we'll shop around for CROs and talk to them to see who has the proposal that makes the most sense for what we're looking for, both budget-wise and claims-wise and time-wise.
That's
because we want to make sure we get the data back in as quickly as possible.
Yes.
So once we've kind of landed on, okay, you're the one who has the vision of what we're wanting, then we start writing the protocol more detailed. They give us a draft. Usually we go back and forth, similar to what we did on the in vitro studies.
Okay.
And so then we kind of lock it into place. But what's different then is the IRB approval, if you want to go into that.
Yes. So once we decide on a protocol, they will take the protocol to a specialized, they call it internal review board, and that could be a third party on their part as well, or they may actually have an internal review board that consists of physicians, statisticians, other scientists, and primarily it's more concerning more about safety issues at this point.
Yeah.
Is blood being drawn, saliva or other bodily fluids? Are we doing biopsies or things like that? So they look at the invasiveness of the whole study.
Is the study going to hurt the
Yes.
consumer, or is it reasonable?
Is an acceptable study, right? And then they either approve it or they reject it, and then it goes back to the CRO, and we sign off on it, and
We can start
we
executing.
can start it.
And when you say acceptable both from like a safety standpoint, nobody's going to be hurt
And
or
a methodology
injured.
perspective
Yeah,
as
the
well.
methodology,
Yes.
like they're reviewing it to say, well, your protocol and what you're trying to measure, the end result, really don't match.
Correct.
Okay.
Yeah. But also from a safety standpoint on what's in the products. Because we have to tell the CRO, we have this in the product, we have these ingredients in the product.
Yeah.
And then they do the safety assessment of those ingredients. Toxicology assessment. And exclusion is pregnant women or, kids can't be, you have to be this age. So there's a lot of steps that they look at. Safety, mostly safety steps that they look
for example,
at.
on the skin side too, before we start a human clinical, we have to have some testing already done. We have to have safety testing, like we talked about 2 weeks ago, the patch testing, the repeat insult patch testing. We have to have that usually completed so that we can show that there's tolerability for the product. And so there's low risk of anyone having any issues within the study. So that kind of lowers the risk right off the bat. We also have to prove that the product is stable and also that it has no micro. So it's very stable as far as micro as well. So that the product that they're getting is as good as a final product and they're not risking anything unnecessarily.
Right, so that's the difference to dietary supplement, to ingestibles, because in ingestibles, they look at the safety of the ingredients. On how safe are the ingredients? Is it caffeine? How much caffeine do you have in there? If you do 400 milligrams of caffeine, they're going to say no, because that's the upper tolerable limit that the FDA has set for safety on caffeine. So they'll say no right away. But if it's 100 milligrams, they'll say yes. But then they'll also look at the herbs that you're testing, right? They may come back and say, show me some safety data on these. So we don't do preliminary safety like you do with the patch test and some of the other testing that you do in topical applications. So they go more into the literature and experience.
Got it. And like duration of these types of tests, like how long does a human study last?
Yeah, so I'll talk skin again.
Yes.
I'll let you talk ingestibles. So like I mentioned, it can be something that's within 1 day, depending on what the metric is. Sometimes we want to see something immediately happening. Hydration, we want to see an immediate radiance. And we're just like, we don't care what it does 12 weeks from now. We just need to we want a 1 hour claim. It can be that simple. Or it can go all the way out to 16 weeks, especially with any sort of wrinkle studies. Anything with pigmentation and age spots.
Yeah.
It takes time for there to be change. And so you tend to go out a little bit farther typically. I mean, it depends on what your ingredients are to ensure that you capture what will happen. I mean, you also put like multiple time points within there. So for example, if you do a 12-week or 16-week study, you're going to have them come in at 3 weeks and 6 weeks because you're hoping, hoping you're going to see data earlier on.
Yeah.
But what you're wanting to show is that like we start seeing benefit at 1 week, but if you keep using it through 12 weeks, 16 weeks, look what will happen.
Yep.
And so it kind of just shows the progression of efficacy.
Okay.
But not just that, and this is the same with ingestibles, is the recruitment takes time too. So if you need 50 people and they trickle in over time, they're not all going to come at the same day. 50 people, you recruit maybe 2 people a day.
Yeah.
Or 10 people a week. So it'll take 5 weeks just to recruit all of the 50 people. And it's the same with ingestibles. So that time frame adds on to when the last data point comes in.
That makes
But
sense.
ingestibles can also go longer too, depending on what they're looking
depending
at.
on what you're looking at. If you're looking at a 1 week or an immediate boost with caffeine, I'll take caffeine as an example. Caffeine, we all know that caffeine is pretty immediate, right? So that may, again, only be a one-time time point as well. Whereas if you do something else, another benefit that you're looking at, that may take 3 or 4 months to do. So it all depends on the end result that you're looking for.
Yeah.
Yeah.
Participation, similar to like the consumer trial, like as far as 30 to 50 participants, or does
Yes.
yeah.
What you're looking for is statistical significance. So it'd be
Okay.
similar criteria.
Yeah.
Yeah.
And so what they'll do is a lot of times we'll give parameters. Like our target audience for this product is age 35 to 55. And we want to make sure for a skin product, we don't want to just target people with incredibly pale skin. We want to look at everybody because we have a wide audience. So we want to make sure to recruit within that as well. So we kind of specify. And one thing they also do with the participants for these studies is they are compensated for participating in these
For their
studies.
time and
Exactly.
everything. Yeah.
Exactly. But
Yeah.
they have no affiliation with us whatsoever. They're just
Yeah.
coming in because they want to try a product.
Right.
And I
Exactly.
think I think it's safe to say the majority of the time they don't know that they're even doing it for
No.
us, right?
Oh,
They don't
no,
know.
we don't brand anything.
No,
Yeah.
They
nothing's
keep
branded.
it incredibly. Usually there's numbers or a formula number.
Yeah.
No one knows.
It's called
what it is.
double-blinded.
Yeah.
You know,
Yeah,
randomized
so
on
the-
what they're taking. So
Yeah. So the
Yeah.
participant doesn't know that
No.
like the product that they're testing would eventually be a LifeVantage product.
Oh, not at all. And they probably never will know.
Yeah.
Sometimes it'll be really funny after a study, like a consumer use, they'll be like, "Can you just tell us like what
Yeah.
I really liked this product. Can I keep using
Can I
it?
keep using it?
We get that
Yes.
a lot.
Can I
'Cause
have some more?
Because a part of some consumer testing is that you'll actually bring them in for focus groups too, where you not only have a questionnaire from them, but you get to hear them kind of discussing. And sometimes then you bring in some branding
Sure.
at that point. But a lot of times you still keep it pretty blind, and
Yeah.
you'll hear them. They'll be like, "So this really like made a big difference with my life. Can you just let me some?"
That's
yeah,
funny.
that's funny.
Cost-wise of a human clinical.
So a human clinical, since we want statistical significance and potential write-up, make a paper out of it or something, publication or something, we want it to be statistically significant. So these studies can run into hundreds of thousands of dollars.
Yeah.
$500,000 is not unheard of.
Yeah.
So they can become very, very, very expensive. Plus the cost of the products,
Yeah.
the cost of the analyses.
Exactly.
And so they can become very, very expensive.
Yeah.
Yeah, so it is definitely the highest
Yeah.
tier as far as
Yeah.
cost.
Which and I think that also helps us, and helps those that are listening, how we make the decision between, okay, we've done a consumer trial, do we need a human clinical
Correct.
or not?
Exactly.
And sometimes it's just a
Matter of
it's just-
priorities.
Yeah.
and just a financial decision.
And what
or
type
a-
of product? Yeah, does it make sense? Will it really bring us a lot of benefit to continue to study it in this way? If it does, then let's prioritize that.
Yeah.
But if we've got what we need and we feel like it's compelling as is, let's just continue forward.
Yeah. Okay, maybe talk about this just a little bit, because I know it's one thing there is an element of with these studies, it gives us the word substantiation. It allows us to substantiate from a claim standpoint what the product is actually doing, and there's always a little bit of, it's normal, right? Some frustration. A consumer will use the product and they have an experience with the product, and they want to be able to talk about it in a certain way and what it did for them. And sometimes we have to say, well, you really can't say that.
Right.
Yes.
Correct.
Connect the dots there.
Sorry, we're laughing about this because we hear this all the time.
Yeah.
And we hear that the dietary supplement's not regulated and, but when it comes down to it, the FDA actually tells us what we can or cannot say under the structure function paragraph that they have. And you can only say certain things that have to do with healthy functions in the body.
So not disease
So
states.
you can never, ever talk about diseases or it cures this or cures that. That's why we also have the statement on the bottles if we say something supports heart health or something like that. We're not allowed to treat any diseases because that's what drugs are for, correct?
Yep.
We're not allowed now of course I forget what the statement is, but if you look on your bottle at the very bottom, on every bottle it says
treat,
prevent, or cure.
Yeah, there you go.
Yes, we can't say any of those words because that only drugs can do that, and therefore it's a disease.
Yeah.
So we can only talk about structures in the body, and we can only support them in a healthy way.
And that's dietary supplements. And then if you
Yes.
go over to
And the
topicals,
skin
it's
is
even trickier
Yes,
because we are
exactly.
not allowed on cosmetics to impact the structure at all in our claims. We have to talk about beautifying and making the skin look pretty.
Appearance.
And it's the appearance of. So you'll see a lot of claims of helps with the appearance of wrinkles, the appearance of this. And I mean, it's almost on every claim. That is kind of a way of softening it a little bit to make sure that we're not talking structure, function. There are different categories in the US called over-the-counter drugs where there have been there's more FDA regulation where we can say more claims just
Yes.
because of all the substantiation that's been provided.
Yeah.
If you use an ingredient at this ingredient between this and this level, you can make these claims.
Yes.
Sunscreen actives are one of those that if you put them in, you can make this claim, or like a skin protectant or things like that.
And in that regard, there is something very similar on the ingestible
Mm-hmm.
side as well. And they're called qualified health claims.
Yeah.
But that's a topic for another podcast.
And I think it's important, right? Because a consumer can use the product and they can have a remarkable experience with the product.
100%.
And we certainly don't want to dismiss that. That is their experience. That was their experience with the product.
We hear so many stories.
Yeah, and those are wonderful, right?
yeah.
They're fantastic, and it's
Yeah,
very
they're
encouraging.
awesome.
But because I think that there becomes this frustration every once in a while. Well, like, this is my experience. This is what I want to say. There is your experience, but there's also what's substantiated, and there's also what within the regulations or the guardrails of what's allowed.
Correct.
And that is actually for all of our benefit and protection, because if we didn't have those guardrails, right, where
People just say anything.
people could say whatever they wanted,
Yes,
right,
exactly.
and then you're dealing
and no one's checking them.
And that's
Yeah.
why the FDA back in the 90s started this whole cliche
Yeah.
and
Yeah.
because people were saying outrageous things.
yeah
Correct.
make
And they're trying to
Yeah,
protect the consumer
Yeah.
as much as they
to
can.
protect the
Yeah,
consumer, to make
exactly.
sure that people aren't misled.
So if you just see the
or-
claims, and
Exactly.
you like the consumer doesn't get to see the claims grid, the substantiation
Oh,
files
that
that
we
we
put
have, because we
together.
put
Yeah.
together
Yes.
really robust substantiation files on our products that back up every single one of our claims. But if there wasn't any sort of checks and balances, people would just say whatever,
Yeah.
and there's nothing in the files.
Yeah.
Yeah.
Yeah. Well, I think the important thing, right, the takeaway here is that there are multiple levels of testing,
Correct.
from in vitro to the human or the consumer studies to actual human clinicals. There are multiple levels of testing, and the nice thing is, the wonderful thing and the admirable thing about LifeVantage is that we use one or all three on all of our products. Like, everything is tested, everything is validated, and everything that we say about the product is substantiated with this scientific method and these scientific tests to ensure that, one, we're really delivering what we say the product is going
Yes.
to do.
Yes,
Absolutely.
we feel incredibly
Yes.
passionate about
Yeah.
that. If we're promising that a product does something, we
Yes.
want to be sure that it does
Yeah,
something.
and-
Because you're looking for relief using something, or you want something to change we don't want you to be disappointed.
Yeah.
We want to
Yeah.
have as much confidence as we can in the products we put out. We want to be proud of what we put out
Yeah,
there.
absolutely.
I mean, that's a big, huge part of our job is that we want to be, when I get up on stage to talk about a new product, I want to be so excited that I can't wait. And I'm sure you've seen it before, but I want to just be bouncing off the walls because I can't wait to share because I believe in it so much.
Yeah.
Yeah.
I don't want to be getting up there and being like, oh, it didn't. Maybe it does this, and just having no
Yeah.
belief in
Yeah,
it.
I'm
Because
the same way.
if we don't believe in it,
Yeah.
well, how are you guys supposed to believe in it?
Yeah.
Yeah.
So we do it first for ourselves,
Yes,
and
absolutely.
confidence, and then we do it for you guys.
Yeah.
Yeah. Okay. This has been fantastic. This has been fascinating. And very, very helpful. And hopefully the hope is, right, it gives a level of confidence to those that you're watching or listening of the level of scrutiny that we go through with literally every product to ensure that it is doing what we say it's doing and doing it in I don't want to get back to a safe way because we talked about that last week, or 2 weeks ago, but we are literally testing everything in one method or in multiple methods.
Yeah, and a lot of times they go hand in hand too, because
Yeah.
we'll have a lot of safety
Yes.
endpoints as part of our efficacy studies as well.
Yeah.
So they go together a lot.
Perfect. So what did I not ask that I should have asked?
Yeah. I think you've asked some pretty good questions, actually.
Because we gave you the
think so.
outline, so we made sure that you asked the
Well,
right.
Yeah.
I have 3 things on my paper: in vitro, consumer, and human.
That seems like a lack of note taking on your part. But no, just,
Fair enough.
Just kidding. Just
Fair enough.
kidding.
Okay, so nothing else?
No, I think we covered most of it.
No, I think we've explained why they're important, why we would look at different ones, the benefits that they bring for each one, that there's not just because something, and I guess something to say, just because something launches with just maybe example for in vitro testing, we just did RNA sequencing, doesn't mean it's any less of a product.
Yeah.
We believe in those products just as much as we believe in the other ones. We're always looking to use our judgment of what's the best way to explore this product and really show its efficacy. And it's going to look different for each product.
Yeah.
And so we will believe in every single product, regardless of what tier we go to, what boxes we check.
Yeah. Perfect.
There you
Perfect.
go. I came up with something. There you go.
Well, thanks for joining us. Thank you to you two. Thanks
Yeah, no
for the
problem.
explanation.
Of course.
Appreciate it. And again, email us at productquestions@lifevantage.com and we'll answer those. And one more time, thank you to you two.
Yeah, thank you.
Both for the conversation and the explanation of the studies, but more importantly, for all of the work that you put in to deliver amazing products for these individuals that are with us tonight.
No
No
problem.
problem.
Thanks.
Thank you.
Have a great evening.
Thanks, guys.
Thank you for tuning in to the Activation Nation podcast. Be sure to subscribe so you never miss an episode. Tell your friends and share your biggest takeaways from today's discussion with anyone who could benefit from them. This episode is sponsored by LifeVantage Legacy, a nonprofit dedicated to improving lives and building a lasting impact for those in need around the world. Learn more at lifevantage.com. We look forward to sharing more with you during next week's conversation. Thanks for listening and being an important part of the LifeVantage community.